Our next section in parasitology is going to be under the phylum Platyhelminthes. And again, this is commonly known as the flatworm category. So the class we're going to be starting with is Cestodes, which are known as Cestodes 1 through 3, commonly known as tapeworms. So we'll find this in chapter 46 of the book, page 302 to 305. And the first 3 cestodes that we're going to cover are Dipylidium caninum, Taenia taeniiformis, and Taenia saginata. So let's start with Dipylidium caninum. This is known as the double-pored tapeworm, and also known as the flea tapeworm. The adult form of Dipylidium caninum has proglottids that look like cucumber seeds. And remember that proglottids are each segment of a tapeworm. So each of these proglottids look like little cucumber seeds, and they also have 2 lateral genital pores on each side of them. This allows the eggs that are located inside of each proglottid to be able to be squeezed out whenever that proglottid contracts outside the body, and those eggs can be squeezed out. Now the adult also has what we call an armed scolex. That means it kind of has teeth or sharp objects coming off of its scolex in order to really anchor into the small intestinal wall. So an armed scolex with a rostellum, and this projectile coming out of the scolex is what we call the rostellum. And again, it is armed. It has teeth or sharp objects on it to anchor it in. Each of the scolexes for Dipylidium caninum also have 4 suckers. So you can see the 4 suckers here in the micrograph picture. From a side view, these circular objects would be the suckers. And again, the adults are found in the small intestine. Now, the larval stage is known as a cysticercoid, and this is what it looks like here in this picture. And you can find the cysticercoid stage in the intermediate host for this parasite. The eggs of Dipylidium caninum are unmistakable. They are oval in shape. They're rather large. You can usually point these out under low power of your microscope right away. And inside of these eggs, there are very defined embryos. And each of these embryos, when this big egg hatches, can actually have the potential to become a cysticercoid larval stage. So we call this a packet of eggs, and it has very defined nuclei. So the DH for Dipylidium caninum is going to be dogs, cats, and occasionally humans. The intermediate host is the flea. The prepatent period is about 2 to 3 weeks for this parasite. So the life cycle of Dipylidium caninum starts with the definitive host. We have the adult form of the parasite in the definitive host, and one of the proglottids from that parasite will break off in the feces. And once it breaks off in the feces, that proglottid will dry out a little bit, and it contracts and releases those eggs into the environment. Now, the eggs are actually ingested by flea larvae. And so the flea larvae, once they develop into an adult, those cysticercoid, the larval stages, will develop inside of them as well. And then the flea is ingested by the dog. As you've seen sometimes when a flea bites your dog, it turns around right away and itches, or it starts biting at its fur and skin, and that's how one of these fleas can be ingested. So once the cysticercoid has been ingested inside of the flea by the dog or the human or the cat, that's when it hatches out of the cysticercoid stage and will become an adult in the small intestine. Now, pathogenesis for this parasite, there really is none. It is considered non-pathogenic. The only possible thing that can occur would be abdominal discomfort, pruritus, which is itching, and also intestinal perforation is possible in very heavy infections. So the next tapeworm we're going to talk about is the cat-wrapped tapeworm, known as Taenia taeniiformis. The adult of Taenia taeniiformis has proglottids that are rectangular and bell-shaped in shape. So here is an example of the cat-wrapped tapeworm here, and I have a red rectangle drawn around one of the proglottids. This isn't here normally. I put it there so you can see the rectangular shape of each of those proglottids. Now, on the side of these proglottids, there is a single genital pore that the eggs will come out of whenever that proglottid contracts, and the adult is found in the small intestine. So the larval stage is known as the strobilocirci. They kind of look like little cysts, and you'll see this in just a minute. You can find the strobilocirci in the intermediate host as well. Now, the eggs, we call Taenia eggs typical of Taniid eggs. And what we mean by that is that there is an embryo in the center of the ova, and there's an outer shell that is thick and radially striated. So when we say radially striated shell, we're talking about it almost looks like rays of sunshine around the entire shell, and that's what we mean by radially striated. So now the definitive host for Taenia taniiformis is the cat, and the intermediate host is going to be the rodent. Now here's a picture of a rodent that has been meat cropsied with this parasite. So the rodent has the larval stage attached to its internal organs. And so this larval stage, the strobilocirci, look like small cysts attached to the organs. So in order to find out how it gets to that point, we look at the life cycle. The Taenia eggs are going to be excreted in the feces inside of those proglottids. The rodents will ingest the proglottids or the eggs that have been spewed out onto the feces, and then they hatch and develop into strobilocirci inside of the rodent. These cyst-form strobilocirci attach to the rodent liver and other internal organs. When the definitive host comes along and ingests the rodent, they also ingest the strobilocirci, that larval stage, and that's considered the infective stage here. So the strobilocirci, once they're inside of the definitive host, mature to adults in the small intestine. Now in cats, again, this tapeworm is non-pathogenic. It is possible in heavy infections, however, to have abdominal discomfort and that itchiness or pruritus. The last tapeworm we're going to talk about today is Taenia saginata, known as the beef tapeworm. The adults of the beef tapeworm, the scolexes, have 4 suckers. They are unarmed, so there are no teeth, there's no rostellum on the scolex here. It's just plain Jane, and so it attaches to the intestinal wall just by the suckers. The proglottids of this particular tapeworm only have a single lateral genital pore, which you can see here in the center picture. The eggs, again, they are typical of Taniids, and so we have that single embryo in the center, the thick, radially striated outer shell, and then the larval stage is known as cystocirci, and it is found in the muscles of cattle. So here is a muscle of a cow, and you can see the cystocirci cyst form in the meat here. Now the definitive hosts of Taenia saginata are dogs and, yes, humans. The intermediate host is going to be cattle, and the prepatent period will be 100 days. So for the life cycle, the dog or the human will shed the proglottids in their feces. Those proglottids will contract, releasing the eggs into the environment, and those eggs can survive for weeks or months in pastures or in sewers located around the cattle. Cattle are infected by ingesting those eggs that are on the contaminated pasture while they're grazing, or in feedlots, hay or silo feed. So for this to happen, that means that farm workers and dogs are defecating in fields or in feedlots. A good farmer will not allow that to happen. You need to set up the right types of facilities to allow those animals and those people to use a restroom so that this is not passed on to cattle and then ultimately on to dogs or humans again. But let's backtrack a little bit. Once the cattle have ingested the eggs on pasture or in the feedlot, the eggs will hatch in the cattle. The embryos will migrate to the skeletal and cardiac muscle and develop into their cystocirci cyst forms. This is the infective larval stage. So dogs and humans are infected by eating raw or undercooked beef. this is how it happens. So once a dog or human ingests that undercooked beef, the cysticerci attaches to the mucosa of the small intestine and will start to develop the proglottids, becoming an adult. So pathogenesis for this parasite in humans, usually non-pathogenic. You may not even know that you have a tapeworm. The clinical signs could include diarrhea and abdominal pain. You will know when that happens. So cattle, this is again non-pathogenic. Having these cysticerci cysts in the muscle actually don't seem to bother the cattle. But whenever the meat from beef cows is inspected at a USDA facility, the USDA officers in charge and inspectors in charge will notice the cysticerci in the muscles and automatically label this measly beef. Whether or not they let it leave the facility at that point is up to them, but usually they don't let it leave the facility. Now, there is one thing to keep in mind, and that is sheep. So if sheep become the intermediate host, meaning the sheep eat the eggs from the pasture or a feedlot, they can actually develop ataxia, blindness, and circling disease. That is because the cysticerci actually head to the brain in sheep, and it can take up this much space. As you can see here in the bottom picture, there is a hole in the brain, basically, where a cysticerci had formed and had grown. So this is why sheep can develop ataxia, blindness, and circling disease when it's taking up that much room in the brain. So that's something to keep in mind for this parasite. So this is Cestodes 1 through 3.